Now let's start with dentinogenesis. Dentinogenesis. Again we are coming to genesis. So what is genesis? Formation. And dentino means formation of dentin. So let's start with dentin formation. That is dentinogenesis. Which cells are involved in the formation of dentin? Blast cells may be involved. So which blast cells? Amyloblast? No, amyloblast will form enamel. Then cementoblast? No, they will form cementum. So odontoblast cells. Which cells? Odontoblast cells are involved in the formation of dentin. So how do odontoblast cells form dentin? They are directly derived from the neural crest cells? No. They are ectodermally derived or ectomesenchymally derived structure? They are ectomesenchymally derived structure. So how is dentin formed? Under the influence during the Bell stage, advanced Bell stage, what happens? Under the organizing influence of inner enamel epithelial cells, here there are inner enamel epithelial cells. Here you can see the columnar cells I am drawing. They are inner enamel epithelial cells. So now, under the organizing influence of this inner enamel epithelial cells, here what is present? Dental papilla. Ectomesenchymal cells of dental papilla are present. So these cells undergo condensation and they form what? Dental papilla. Now the peripheral cells of dental papilla under the influence of inner enamel epithelium differentiate into what? Odontoblast cells. So here we can see formation of odontoblast cells. This peripheral cells, undifferentiated mesenchymal cells, differentiate into odontoblast cells. So now under what? Under organizing influence of inner enamel epithelial cells, that are amyloblast cells, undifferentiated mesenchymal cells, undifferentiated mesenchymal cells. Chymal cells of what? Of dental papilla. DP, again DP. D will form DP, dental papilla, is involved in the formation of what? D for dentin and P for pulp. So undifferentiated mesenchymal cells of dental papilla, not dental sac, dental papilla, will differentiate into, under the influence of inner enamel epithelium, they will differentiate into which cells? Odontoblast cells. Now this odontoblast cells will be involved in formation of what? Dentin. So dentin is found only in crown portion or in root portion as well. Dentin is found in crown as well as root. In crown it is covered by enamel and in root it is covered by cementum. So here odontoblast cells will form dentin. So once dentin is formed, then enamel formation will start. Now this is the basic formation of dentin. In case of root dentin, how is root dentin formed? Slightly different mechanism. Now here you can see in this diagram inner enamel epithelium, and here you can see outer enamel epithelium, right? So this junction is known as cervical loop. Cervical loop, where inner enamel epithelium and outer enamel epithelium fuse. So this portion has only two layers, inner enamel epithelium and outer enamel epithelium. This layer further proliferates. So inner enamel epithelium and outer enamel epithelium proliferate. And they form what? HERS. Again, role of HERS. HERS is involved in root formation, so it is also involved in formation of dentin. So HERS, Hartwig's epithelial root sheet. Now this cells, here you can see this is HERS. Separates dental papilla from what? Dental sac. They separate dental papilla from dental sac. So here you can see dental papilla, and here you will see dental sac. So dental papilla is separated from dental sac. Separates dental papilla from dental sac. Now here there will be inner enamel epithelium, and here there will be outer enamel epithelium, right? Only two layers will be present in HERS. So under the influence of again inner enamel epithelium, dental papilla cells will differentiate into odontoblasts. So again in HERS, under influence of inner enamel epithelial cells, dental papilla cells differentiate into which cells? Odontoblast. And this cells then start formation of what? Radicular dentin. Radicular dentin. So this way radicular dentin is also formed. But in radicular dentin, this dentin will not influence the inner enamel epithelial cells, and they will not differentiate into amyloblast cells. So enamel formation is not there in root, only dentin formation is there, and then cementum is formed. So remember, in crown they will have reciprocal inductions. What is reciprocal induction? Under influence of amyloblast, dental papilla differentiate into odontoblast, and this odontoblast will form first layer of dentin. So when first layer of dentin is formed, then it again influences the amyloblast cells to form enamel. So here we have reciprocal induction. Both are influencing each other's formation. Okay. So crown dentin, we have completed formation, and root. Root, mainly HERS is involved in its formation. It separates dental papilla from dental sac, and under the influence of inner enamel epithelial cells of HERS formed by the proliferation of inner enamel epithelium and outer enamel epithelium from cervical loop, right? So they influence the surrounding dental papilla cells to differentiate into odontoblast cells, and then they form radicular dentin. So in this way dentin formation is started. Now dentinogenesis is a two-phase sequence, right? Just as enamel matrix formation, then mineralization. So here also, but difference is in enamel, after matrix formation, immediately mineralization will start. But in dentin, sufficient amount of organic matrix will first be deposited, then it will wait for some time, and then mineralization will start. Okay, so coming to dentinogenesis. It is a two-phase sequence. Two-phase sequence. It involves what? Two-phase means first one will be matrix formation and the other one will be mineralization. Once matrix formation will be, when matrix will be deposited, then only mineralization will start. So first is matrix formation. And the next is mineralization. So how is matrix formed? Collagen fibers may be present. So how will they be deposited? So starting with collagen type 1, and precursors of porphyrin. Precursors of phosphorin. Precursors of phosphorin are present in organelles, cell organelles, such as endoplasmic reticulum, Golgi apparatus. So now collagen type 1 and phosphorin are present in organelles, cell organelles. Then which cell? Odontoblast cells. Now we have seen odontoblast cells have extensions. They have what? They have extension. This extension is known as cytoplasmic process. So they will migrate in its processes. Migrate in processes. Cytoplasmic processes. Then they are emptied in where? Matrix. Extracellular matrix. From the process they are emptied in extracellular matrix. So once they are emptied, here starts the deposition of organic matrix. So from the cell organelles, they are transported to the processes, then from the processes to the extracellular matrix, and here the deposition of collagen matrix takes place. So matrix formation begins here. Matrix formation begins with, we've already discussed, begins with what? Deposition of which fibers? Small diameter or large diameter? Which fibers? Small diameter fibers or large diameter fibers? Large diameter fibers. And these fibers are known as, this was an MCQ question while discussing, I told you. So these fibers are known as, large diameter fibers are Von Korf's fibers. Which fibers? Von Korf's fibers. So matrix formation begins with large diameter collagen fibers, and these fibers are known as what? Von Korf's fibers. Von Korf's fibers. So they're laid perpendicular or parallel to DEJ? Perpendicular. They're laid perpendicular to DEJ. Laid perpendicular to DEJ. It has appearance of corkscrew appearance. Which appearance? Corkscrew appearance. So first form dentin is known as what? Mineralized first form dentin will be mantle dentin. It is less mineralized than circumpalpal dentin. But so once mantle dentin is formed, then collagen fibers, rest of the collagen fibers will be laid parallel, not perpendicular. They will lay parallel. So once mantle dentin is deposited, rest of the collagen fibers, will be parallel to what? DEJ. So first laid fibers will be perpendicular and the others will be parallel. Remember this. So it is a two-phase sequence. It involves what? First, matrix formation, and second, mineralization. So matrix formation starts with what? Collagen and precursors of phosphorin are present in organelles. From there they will be transported to cytoplasmic processes of odontoblast cells and then to the extracellular matrix. So here collagen deposition finally starts. So matrix formation begins with deposition of which? Large diameter fibers, and this large diameter fibers are known as Von Korf's fibers. What? Von Korf's fibers. They are laid perpendicular to DJ, not parallel. So once mantle dentin is formed, the rest of the collagen fibers will be laid parallel to DJ. Now coming to mineralization. So mineralization, we all know, deposition of crystals will be started here. So how? As the odontoblast give their processes, matrix vesicles bud off. So we have seen in mantle dentin, mineralization is by matrix vesicle, remember? And in circumpalpal dentin, it was by heterogeneous enucleation. So I had also explained what is heterogeneous enucleation. They have nucleators, template for mineralization. What are nucleators here? Non-collagenous proteins. So first mantle dentin is formed or circumpalpal dentin is formed? First mantle dentin is formed. So mineralization, how does mineralization take place here? By matrix vesicle. So we will study as odontoblast give their processes, matrix vesicles bud off. As so they contain what? They already contain alkaline phosphatase. So they contain alkaline phosphatase. Now this combines with calcium that is taken up from the fluid into the vesicle. So combine with calcium taken up from the fluid into the vesicle. Calcium. And form what? Apatite crystals. Crystals. So then they grow, right? They will grow inside the vesicle. They will grow. As they will grow, then they will rupture the vesicle. They will not further be present in this vesicle. This is the closed vesicle, and if this is the vesicle, here the crystal starts growing and it will rupture the vesicle. So this is vesicle. Here our crystals are present. It will grow in size and finally it will rupture. So it will grow and rupture from vesicle. From vesicle. Now once they rupture, same way another clusters of crystals may be formed. Here one crystal, here one crystal, and here. Then these crystals will finally fuse together and form a uniform matrix. This cluster, they will fuse and form a uniform matrix. So they will fuse with adjacent clusters. And form a uniform matrix. And if they fail, we have already discussed which dentin is formed. If fusion has not taken place properly, then what is formed? Which dentin? Interglobular dentin will be formed. So defect will be in mineralization, not in matrix formation in case of interglobular dentin. If the fusion is not proper, then we will see interglobular dentin. And form uniform calcified matrix. And next, further matrix will be formed by non-collagenous proteins. So they will mineralize by heterogeneous enucleation. So which were the non-collagenous proteins? Dentin, CLO protein, dentin phosphoprotein, chondroitin sulfate. We have already discussed in composition of dentin, chemical properties of dentin. Now what is the pattern of mineralization? One we have seen, globular. So only one pattern of mineralization? No, it might have as well linear calcification. So coming to the pattern of mineralization, you might not be having any doubt in this two-phase sequence of dentin formation. First is matrix formation and the other one is mineralization. So matrix formation, collagen and phosphorin are present in where? Organelles. They will enter the cytoplasmic process and then they will be deposited in the extracellular matrix. And then larger diameter fibers will be formed and they are known as von Kopff's fibers, perpendicular to DJ, and rest of the fibers parallel to the DJ. So next, coming to mineralization. So in mineralization, what will happen? As the odontoblast give processes, matrix vesicles bud off. These vesicles contain already alkaline phosphatase. This then combines with calcium taken from the fluid and it will form hydroxyapatite crystals, and then they will grow inside the vesicle and rupture the vesicle, leading to fusion with the other clusters and form a uniform matrix. This is for first form matrix. Then for circumpulpal dentin, that is the next form dentin which forms the bulk of the tooth, mineralization takes place by heterogeneous enucleation. Non-collagenous proteins will be involved in mineralization. For mineralization of rest of the dentin, mineralization of rest of dentin, non-collagenous proteins will be involved. And these proteins we have already discussed. So now coming to pattern of mineralization. In matrix vesicle and at mineralization front, in matrix vesicle and mineralization front, we will have globular pattern. We will have globular pattern. So in globular pattern, what happens? We have already discussed. Clusters will be formed and then they will fuse together. And in case of circumpulpal dentin, globular pattern if deposition of dentin is fast and linear if deposition is slow. So circumpulpal dentin will have two patterns: globular if deposition of dentin is fast, dentin is fast and linear if deposition of dentin is slow. . is slow. Now, which genes are involved in the formation of dentin? Genes involved in formation of dentin. Genes involved in formation of dentin. So in formation of dentin, odontoblast cells plays an important role. So first step will be odontoblast differentiation. So which gene will be involved? MAP1B. This gene is involved in odontoblast differentiation. And the other one is PHEX, PHEX. So once odontoblast differentiate, they start forming dentin. So collagenous matrix will be laid down, and the next step is mineralization of that matrix. So PHEX gene will be involved in mineralization of dentin. So we have discussed the genes involved in formation of dentin. Which genes are involved? MAP1B, MAP1B, odontoblast differentiation, and PHEX, dentin mineralization. Now coming to clinical considerations of dentin. So clinical considerations will be dentinal caries may be there, right? Then sensitivity, we have studied the theories for sensitivity of dentin. So dentin will have sensitivity as well. Dentinal sensitivity can be also present. Then caries, sensitivity, age changes also we have seen, translucent dentin, dead tract formation. Then we will have dentinogenesis imperfecta, same as that seen in amylogenesis imperfecta. Dentinogenesis imperfecta, word only says dentinogenesis, formation of dentin, imperfecta. So something disturbance during the formation of dentin leading to defective dentin formation. So dentinogenesis imperfecta. It might be having hypoplastic dentin, hypomineralized dentin, okay, or hypocalcified dentin. The next is, it may be associated with osteogenesis imperfecta or may not be associated with osteogenesis imperfecta. Dentin might appear brownish or amber colored in dentinogenesis imperfecta. The next coming to dentin dysplasia. Here, rootless teeth may also be present. Dentin dysplasia also known as rootless teeth because there might be resorption of root dentin. So, dentin dysplasia. So, these are the clinical considerations. So, here coming to the diagrams of dentin. Dentin formation is shown here. These are amyloblast cells, here you can see reversal of polarity. If inner enamel epithelial cells will be there, then it will have nucleus towards the basement membrane. But when they are differentiated into amyloblast cells, you can see reversal of polarity, that is nucleus away from the basement membrane. Here you can see this is the basement membrane and nucleus is away from the basement membrane. So these are amyloblast cells. Then this is dental papilla. Their peripheral layers will differentiate into odontoblast cells. Which cells? Odontoblast cells. These are odontoblast cells. So under the influence of amyloblast cells, this dental papilla cells differentiate into odontoblast cells. Here you can see odontoblast cells. And these cells will start formation of dentin. This is formation of dentin has started. So you might not be having any doubt. Amyloblast cells, dental papilla cells differentiating into odontoblast cells. These are odontoblast cells. Okay. Now mantle dentin. Here, as you can see, this is first form dentin. Collagen fibers are larger and perpendicular to DEJ. Calcification pattern is mainly globular. So here you can see globular pattern of calcification. And they fuse. Here you can see 1, 2, 3 globules, and this globule will fuse together and form dentin. Then this is linear calcification. If deposition of dentin is slow, not fast. If deposition of dentin is slow, so in that case, linear deposition, linear calcification. Dentinal tubules were already discussed. Then predentin also we have discussed. Neonatal line, you can see here, same as in dentin. Here you can see neonatal line in dentin, differentiating prenatal enamel, pre-birth enamel from pre-birth dentin from post-birth dentin, same as seen in enamel. Pre-birth enamel was differentiated from post-birth enamel. Same way here, neonatal line differentiates pre-birth from post-birth. And better quality was pre-birth dentin because it is in a protective environment. It gets sufficient nutrition, sufficient nutritional supply and environmental favorable environment for the growth, right? And after birth, there might be some disturbance, so leading to accentuation of incremental lines, which is neonatal line. Interglobular dentin, failure of fusion of globules, leading to interglobular dentin. This you can see here, fusion has not taken place. Here also fusion has not taken place. So we can see interglobular dentin. Here dentin is formed, but here interglobular dentin will be present. And it will be present where? In mantle dentin or in circumpulpal dentin? It will be present in circumpulpal dentin, just beneath the mantle dentin. Tom's granular layer. So here you can see granular layer formed by interlacing and looping of dentinal tubules. This is Tom's granular layer. Dead tracks. Dead tracks. You can see this are empty dentinal tubules formed because of retraction of odontoblastic process or death of the odontoblast cells. So they are filled with air and they will appear dark. So we have completed with topic dentin. If you have any doubt, you can contact us. you can post in our Telegram group, and we are there for you. Again and again, I am reminding you. So please start studying, then only you will have doubts, right? So we have completed with dentin. Dentinogenesis, dentin formation, types of dentin, and fibers of dentin. Physical and chemical properties of dentin, as well as its clinical consideration. Theories of dentin sensitivity were direct neural stimulation theory, transduction theory, and fluid or hydrodynamic theory. And the most widely accepted theory was fluid or hydrodynamic theory. Right? Collagen fibers, please remember, in mental dentin, larger diameter fibers, worn corpse fibers, and they are mainly type 3 collagen, and the rest of the dentin collagen fibers will be parallel. So we are completed with dentin.